So welcome back, guys. Today we will going to discuss about a topic that we have not discussed earlier, that is DNA separation by equilibrium density gradient centrifugation. So it is nothing but just a application of the isopycnic centrifugation that we have discussed earlier times. So here again we are constructing a gradient of density, okay, and the density will be increased from the top to bottom. So here the thing that we are using to create the density gradient is CSCl, that is cesium chloride. So why we are using cesium chloride to separate DNA molecules? So we are using cesium chloride to separate DNA molecule because cesium chloride have a similar density at a concentration of 1.6 to 1.8 gram per milliliter, okay, and that is useful to use to separate DNA molecule. So as we are separating other things with the help of isopycnic centrifugation, here we are also using the same property for separation of the DNA. That means here we are using the Bowen density, the differences in Bowen density between different DNA molecules to separate them, okay. So as it is a isopycnic centrifugation type, so it is obvious that the density gradient will form during centrifugation, okay, and we know that in case of red zonal centrifugation the density gradient will be preformed before centrifugation, right? So here the density gradient centrifugation will be formed during centrifugation, okay. So how the DNA molecules differs in their Bowen density? So different DNA molecules have different relative proportion of ATGC, it is obvious, right? So remember that the molecular weight of AT base pair is lower than the GC, okay. Now try to think about this, then it is obvious that the which DNA molecule have a higher proportion of GC have a higher molecular weight, right? Similarly, the DNA molecule who have a higher proportion of AT have a lower molecular weight, okay. So in turn we can say that the molecule who have a higher molecular weight should have a higher Bowen density, right? Because Bowen density is nothing new, it is density in that particular solution, right? So and we all know that density depends on the molecular weight. So that means the DNA, if we consider two DNA molecule, let's say this is one and another one is, I am drawing with a green color, okay. So now among these two DNA molecule, in case of the red one the GC content is 40%, and in case of the green one the GC content is, let's say, 60%, okay. So it means if it is a DNA A, if it is DNA B, then the DNA B have a higher molecular weight and higher Bowen density than the DNA A, right? Am I right or wrong? Right? So that is the property that here we are using to separate them on the basis of their Bowen density in the isopycnic centrifugation. That is here we are mentioning as equilibrium density gradient centrifugation, okay. And here we are using cesium chloride to construct the density gradient, okay. And why we are using cesium chloride? Because of the concentration 1.6 to 1.8 gram per milliliter of cesium chloride have a similar density with that of the DNA, okay. So that is the idea why we are using DNA for, why we are using cesium chloride for separation of the DNA, and why we are using isopycnic centrifugation, okay. Now there is a relation between the density of the DNA with their GC content, okay, and it is a kind of linear relationship, that is rho here we are using for density, rho is equal to 1.660 plus 0.098 into G plus C gram per centimeter cube, okay, where G plus C is the molar fraction of the G plus C in the double stranded DNA, and rho is the Bowen density of the DNA. Okay, and now if we try to plot this in a graph by taking rho at the y-axis and x as the percentage of base pair that in GC in x-axis, then we will get a linear curve. That simply means rho is directly proportional with the percentage of GC base pairs, right? That is why it is constantly increasing with increase in rho. Okay, so up to that point we have learned that we are used to separate DNA molecule or to fractioning DNA molecule the density with the help of the density gradient centrifugation. And here we are using the isopycnic type of density gradient centrifugation, where the gradient will form during centrifugation. Okay, and here we are preparing the density gradient by taking cesium chloride. Okay, so cesium chloride have a lower density in the upper part of the tube and have a higher density in the bottom of the tube. Okay, and why we are using cesium chloride? We are using cesium chloride because cesium chloride have a same density to that of DNA at a concentration of 1.6 to 1.8 gram per milliliter. Okay, and how we are separating DNA on the basis of their Bowen density? Because if we consider two DNA molecule, so they should have differences in AT and GC. Okay, and another thing I want to mention here, if the differences is GC content is greater than 5%, then they will appear as a two different band in the tube. Okay, otherwise it becomes very hard to distinguish, so that you may consider as a drawback. Okay, that you will need at least a 5% differences in your GC content so that you can separate two DNA sample. So now here a interesting information, that now if we run a sample in a density gradient centrifugation which consists of linear double stranded DNA, single stranded DNA and single stranded RNA, then you will get band for linear double stranded DNA at the bottom. Okay, and you will get a band for single stranded RNA at the middle, and single stranded DNA will be at the top. Okay, and another interesting fact here, that you will never get any kind of pellet here, because we are talking about isopycnic centrifugation, and in the isopycnic centrifugation explanation I have told you that isopycnic centrifugation you don't get any kind of pellet. Okay, and it is independent of time. So the Bowen density of most of the cases double stranded linear DNA is 1.70 gram per centimeter cube in cesium chloride solution, and density of the single stranded DNA is 0.015 to 0.020 gram per centimeter cube, and the Bowen density of the single stranded RNA is less than greater than 1.8 gram per centimeter cube. Okay, so that means in the test tube, if we run the sample containing linear double stranded DNA, single stranded RNA and single stranded DNA molecule, then according to this data we will get single stranded RNA at the bottom, then we will get the linear double stranded DNA at the middle, and then the single stranded DNA. Okay, so this is the correct information. So the previous thing that I have provided which is wrong, so you have to remember about this one, that single stranded RNA who have a higher Bowen density than the linear double stranded DNA and then the single stranded DNA. Okay, so now we will going to see some application of DNA separation with the help of this equilibrium density gradient separation. So first important thing is the separation of satellite DNA from the rest of the DNA. Okay, so what is satellite DNA? Satellite DNA is the DNA molecule which have a highly repetitive sequences. And highly repetitive sequence means it have either a increase in GC content or either a increase in AT content, right, depending on which is repeating. So it is obvious that the satellite DNA will have a difference in their Bowen density than the rest of the DNA molecule, right? So we can also use this Bowen density centrifugation technique or equilibrium density gradient centrifugation technique to separate satellite DNA or rest of the DNA. Okay, so after centrifugation we will get two band, one for rest of the DNA and another one for satellite DNA, but it is not for all the cases. There can be some situation when the average base composition of the satellite DNA is quite equal to the rest of the DNA. Okay, under such circumstances it becomes very difficult to separate satellite DNA and rest of the from the rest of the DNA with the help of equilibrium density gradient centrifugation. Okay, so here we have taken an example of mouse DNA. We are using a fragment of DNA taken from mouse and we are centrifuging them in a CSCL density gradient centrifugation. So what we will get? We will get two band. One band consists of 8% of the DNA with 30% GC content. This band is for the satellite DNA and another one is 92% of the DNA containing band with 42% of the GC content. Okay, and this is the band you will get at the bottom. Okay, so that is how we can separate satellite DNA from the rest of the DNA with the help of density gradient centrifugation. Remember it very carefully that the concept is same. We are using the differences is base composition, right? And GC content have a higher molecular weight than the AC content, and that is nothing new, right? Not AT, it is AC, not AC, it is AT, right? So as GC have a higher molecular weight, it is clear that they will have or they will provide a higher Bowen density, right? So that is the concept that will you may observe in every cases. So now another important thing that I want to discuss, that this is another application of cesium chloride density gradient centrifugation, that is you can separate supercoiled circular DNA molecule from the linear double stranded DNA with the help of this kind of cesium chloride density gradient centrifugation. Okay, but under such circumstances or when you are performing this kind of experiments, you have to use ETBr, that is the ethidium bromide. So ethidium bromide is an intercalating agent that we have to use here. Why we are using ETBr? So remember, ETBr will unwind all the helix that is present. Okay, because we all know that DNA is a double helical structure. So for doing the experiment very well, it is necessary to unwind them. So for unwinding of the double helix we are using ETBr. Okay, so not only ETBr will help in unwinding of the double helix, but it will also reduces Bowen density. Okay, that means when you are using ETBr and you are performing a density gradient centrifugation, the Bowen density you will get is not the actual Bowen density because ETBr already reduced them. Okay, so now in case of circular supercoiled DNA molecule, there is a very little chance for ETBr to bind because circular double stranded circular supercoiled DNA molecule is closed from every end, right? That is, I have written here, that is circular covalently closed circular supercoiled DNA molecule has no free end. Okay, and as a result ETBr has a very little freedom to unwind. Okay, and can only allow a limited amount of ETBr to bind, and that is why in case of covalently closed circular double stranded DNA molecule, there will be a very small decrease in the Bowen density. that is as much as 0.085 gram per centimeter cube. Okay, that means in case of covalently closed circular double-stranded DNA molecule, as there are no free end present, it becomes very difficult for ETBR to bind and facilitating the unwinding. Okay, and not only that, as here very small amount of ETBR can bind, it also reduces the density at a very small amount. Okay, near about 0.085 gram per centimeter cube. Okay, and that is why during this kind of separation, remember it very carefully that the covalently closed circular supercoiled DNA molecule or any kind of DNA molecule which is supercoiled will have a greater Bowen density. Okay, but in case of linear DNA molecule which is not topologically constrained, there is a chance of larger ETBR molecule can bind with it, right? And as a result, there will be a significant decrease in Bowen density in case of linear DNA molecule. Okay, that is as much as 0.125 gram per centimeter cube. Okay, so that is why you will get linear DNA molecule at the top of the tube. Okay, that means the band you are getting at the top will be for linear double-stranded DNA molecule. Next circular ETBR means there is a particular position where the ETBR can find a gap. Okay, and they can access this gap to increase their binding and unwinding. That is why next circular double-stranded DNA will appear at the bottom and supercoiled DNA will appear. You will get next circular DNA molecule at the middle. Why I have explained you that in the next circular DNA there will be a little gap and present. This little gap will allow a relatively larger amount of ETBR to bind. Okay, and there will be a relatively higher unwinding as compared to the supercoiled DNA molecule. Okay, and that is why you will get a band for next circular DNA molecule at the middle. Okay, and as supercoiled DNA is completely closed from every end, very small amount of ETBR can bind and which will facilitate very small amount of unwinding. Okay, and that is why there is also a very less decrease in Bowen density. That is why they will appear at the bottom. Okay, and that is how with the help of Bowen density you can also separate linear double-stranded DNA molecule, next circular double-stranded DNA molecule and covalently closed circular double-stranded DNA molecule. Okay, and under such circumstances you have to use ETBR, remember, to unwind them. Okay, and here we are separating satellite DNA molecule from the rest of the part. Satellite DNA molecule will usually appear at the top and DNA molecule, that is rest of the DNA molecule, will appear at the bottom. Okay, and but this is totally on the basis of the GC content that is present. And here we have discussed the relationship between the density and the GC content, which is a proportional relationship. That means the percentage of GC increasing, the rho will be increasing linearly, right? So that is all about the equilibrium density gradient centrifugation for separation of the DNA molecule. Okay, so for a quick recap, we are using equilibrium density gradient centrifugation technique for separation of the DNA on the basis of their Bowen density. Okay, and here we are constructing the density gradient with the help of cesium chloride. So why we are using cesium chloride? Because cesium chloride have a similar density at a concentration of 1.6 to 1.8 gram per milliliter. Okay, that means the concentration of 1.6 to 1.8 gram milliliter cesium chloride have a density which is equal to the density of the DNA molecule. Okay, now here we are using a kind of isopycnic centrifugation. That means the density gradient will be forming during the centrifugation. Okay, and here we are separating DNA because there will be a relative proportion of 80 GC content in every DNA, right? So the DNA molecule who have a higher AT content should have a lower molecular weight than lower density, and the DNA molecule who have a higher GC content who should have a higher molecular weight and then should have a higher density. Okay, and why is this so? Because there is a 3 hydrogen bond present between G and C. On the other hand, A and T, in between A and T there is only 2. Okay, so there is a relationship between rho and GC, that is our Bowen density and GC, and that is a proportional relationship. That means DNA molecule having a higher GC content should have a higher Bowen density, and it will appear lower at the bottom. Okay, and now we have separated first the linear double stranded DNA, our single stranded RNA, and the single stranded DNA molecule, and we find this that single stranded RNA will be appear at the bottom, then linear double stranded DNA at the middle, and single stranded DNA will be at the top, right? Similarly, we have here separated satellite DNA from the rest of the DNA, right? And depending on the GC content, the appearance will be occurring, and you will get two different band. But remember it, if the GC content difference is less than 5%, so it will be very difficult to separate, right? That is why the DNA molecules, the satellite DNA molecule whose average base composition is similar to that of the rest of the part of the DNA molecule, are very difficult to separate with the help of equilibrium density gradient centrifugation. Okay, and here we have also considered an example where we are separating the satellite DNA of a mouse from its rest of the DNA. Okay, and here satellite DNA will be appear at the top and the rest of the DNA will appear at the bottom. And here we have separated linear double stranded DNA molecule and covalently closed circular double stranded DNA molecule. And remember, in that particular application we have used ETBR to unwind the helix, right? Not only that, the ETBR DNA complex will have a lower density than usual. Okay, and the decrease in density will be lower in case of supercoiled circular double stranded DNA molecule, because supercoiled DNA allow only very small amount of ETBR to bind as compared to the linear double stranded DNA, which is not topologically constrained and allow too much ETBR to bind. And as a result, what will happen? It will observe a greater decrease in Bowen density. Okay, and as a result it will appear in the top. Next circular ETBR DNA complex will appear at the middle, and supercoiled DNA ETBR complex will appear at the bottom. That is the separation scheme. Okay, so that is the concept for today. So today we have discussed about equilibrium density gradient centrifugation based DNA separation. So I hope this video will be helpful to you to understand the concept. So thank you for listening to this class.